Yes, it's Uncle Jegs. Last time we saw the different human defense systems that protect us against pathogens. This video is going to focus on what you need to know for vaccination, and if you have a look at the skills box, we also need to be able to evaluate the global use of vaccine in preventing diseases. So I'll show you how to answer evaluation questions as well. In a future video, I'll dive into the ideas of this brother here and how those ideas led to the mad idea that vaccines cause autism. Well then, let's chop some science. Here are the retrieval questions for this lesson. You know what to do. Pathogens are microbes that cause a disease. Possible pathogens are bacteria like Salmonella, fungi like Rose black spot, viruses like HIV, and protists like malaria parasite. For the last question, it is the surface proteins, the antigens, that allow cells to recognize their ops. A quick recap from the last video. Click the card if you need to check that, but you can probably get the gist of it from these three pictures if you're not bothered to switch over. This activity is asking you to use these three images to describe the actions that white blood cells take in order to protect your body against pathogens. White blood cells engulf pathogens in a process called phagocytosis. White blood cells can also produce antibodies, and finally, white blood cells can produce antitoxins. Following on from the third line of defense that we saw in the last video, antibodies are part of the specific immune system.
How does it remember? Does it have a brain?
That's a good question. To answer that, let's jump into the human body itself. Every now and then, a pathogen or a group of pathogens can make their way into your body.
Ladies and gentlemen, good day.
Some pathogens are more active on the scene than others, but you can see here that each pathogen has its own unique balaclava covering its face. This corresponds to the surface proteins, the antigens on the pathogen. The word antigen literally means antibody generator, so antigens cause antibodies to be produced. Your white blood cells are always on the front lines of this, and it's on sight as soon as they clock a foreign antigen.
Yo bro, where are you from? You're not from the borough, you're not from the area, you're not from the ends.
Once the antigen on the pathogen has been identified, a specific type of white blood cell backs out the proteins and starts spraying out antibodies. When we look closely at antibodies, we see that they are Y-shaped proteins. The tips of it are the antigen's binding site. This region is very similar to the active site on enzymes, meaning that they only allow a specifically shaped molecule to bind to it. In enzymes, that was your substrate molecules, but for antibodies, that's going to be your antigen. And as we saw, different pathogens have different antigens. So the antibody that would be needed for the measles virus will be different from the antibodies you need for the Rona. The white blood cells that scored the point against the specific pathogen becomes a certified OG and becomes a memory cell. Before speaking about vaccines, have a go at these challenge questions. Antigens are proteins found on the surface of cells and pathogens. In the top image, we can see that antibodies help clump up pathogens together, making it easier for phagocytosis to occur. The last question directs you to the image that shows the different antigens. Since viruses mutate very rapidly and evolve rapidly, they will develop different antigens, and so you need to develop different antibodies for each new specific strain of the virus. Make sure you can pattern the implications in this graph. It's a pretty popular graph in this unit. The graph shows the concentration of antibodies in the blood after the first and second encounter with the same pathogen. We have concentration of antibodies on the y-axis and the day on the x-axis. The first challenge question is asking you to read the highest peak on the graph. The second is asking you to state two differences in the line after the first and second encounter. I'll give you a hint here. Use the differences in the axes to help you in your description. The final question is pretty self-explanatory, so have a go at those. Each small square represents 0.2, and that is the peak here. And the peak here obviously must be 5.5. The two differences are that the concentration of the antibodies produced is higher in the second response, and the production is also quicker, or you can say the opposite for the two marks. Finally, the white blood cells that cause the second response is our OG memory cell. What about if the pathogen kills you or deforms your body during the first encounter? What do you do then? That's when you get some help from your doctor.
Stop it. Get some help.
These three pictures show you the process of how we mitigate against such outcomes. Unfortunately, you got to get juked.
Can't we just drink or eat the vaccine?
That's a very good question, but if you remember from the enzymes practical video, your stomach and small intestine contain proteases which break down proteins. These enzymes will break the shape of your antigen before it can get into your blood and cause immunity.
Allow that.
Here are two challenge questions that will set you up for the definition of vaccine, so have a go at that. You can see that the pathogens have been either killed or are now weakened.
Weakness of any kind completely revolts me.
What that means is that they are now no longer pathogenic, so they can't cause a disease, but they still have their antigens intact, so we can use them to generate antibodies by our white blood cells. This is the equivalent of pulling off the belly from our pathogens earlier and using that belly to train your immune system that if anything like this comes into your body again, it's on sight. Despite the effectiveness of vaccines, there are some people that have either very weakened immune systems or they have a reaction to the vaccine, but this is a very small proportion of the population. For these individuals, they are advised not to take the vaccine. This image and the mega challenge here will help you understand how we keep such people safe. Pause and try and understand what's going on here. On the right, you see that a large population that are immune, these are the people that are yellow, mean that the contagious people, which are the people in red, are unable to pass the infection easily to the susceptible people, which are the people in blue. So when a large population of people have been immunized, the people that are contagious or infected are unable to pass the infection. This prevents others that are susceptible from catching the disease. That's how you get your 3 marks there. Well, look at that. We've covered this side of the specification, but now we have to go over how to answer evaluation questions in science. Here's a good opportunity to stress what the difference is between an explain question and an evaluate question. Let's first look at what an explain question looks like. This is a 6-mark question on fractional distillation. The first sentence is setting up the scene for you by letting you know this is about the process that separates crude oil. The command word starts here. Explain how crude oil is separated into different fractions by fractional distillation. When you are asked to explain, you have to make something clear or state reasons for something happening. The marks are segmented like this. To get the highest marks, you have to give logical links with your points. The easiest way to do this is to use a conjunction like because. Look at these different points that you could have put in your answer. I can logically link these using conjunctions. Hydrocarbons vaporize because the crude oil is heated. Vapors enter the fractionating column near the bottom because the column is hotter at the bottom and cooler at the top. These are some examples that you can use. So you basically give a point and then you give an explanation. Now let's look at what an evaluation question looks like. Here we have the question about life cycle assessments. There's a lot of information here setting this up for you, but it's the command words that we are going to focus on. It's asking you to evaluate this statement that's on top. Use your knowledge and the information from the above table to help you. Look at how the marks are segmented here. To get the highest marks, you should have a logically structured evaluation that links several comparisons, but your points also need to be relevant. I recommend starting with the pros and then moving on to the cons. And look how the numbers are not just copied and pasted. The numbers are showing the comparison. For example, instead of saying paper uses 1.7 megajoules of energy and plastic uses 1.5 megajoules, you should state the difference and evaluate whether that's a pro or a con. So I say in the con section that paper requires 0.2 megajoules more energy than plastic bags. There's just one more thing you have to do with your evaluation questions, and that is to give a conclusion. You either have to agree, disagree, or state that the data is inconclusive. That just means that you don't have enough information to give a justified conclusion. Here's another example of an evaluate question. Notice how they haven't given you as much information here because you're expected in the specification, you're expected to know this information without any hints at all. Spend some time reading this question and attempt to give a logical structure of how you should write this out. It's okay if you don't know the information. We are just focusing on the skill that you need at the moment. The question primes you into thinking about CHD, coronary heart disease. So this is all about the heart and the blood vessels, and more specifically, it's priming you to talk about the treatment of CHD. The command word tells us to evaluate the use of statins and stents. That means that we give the pros and the cons for each, okay? And this is how I've laid it out here. I first start with the pros for statins, and then I move to the cons of statins. Then I move to the pros of stents and the cons of stents. And finally, you give your own conclusions. Since you are comparing two treatments, you should justify which treatment you think is the best and why. And there we go. Lesson is done. We looked at how antibodies work and the types of cells that make them. Then we looked at the differences between the first and second encounter with a specific antigen, and also how vaccines take advantage of this. Then we looked at herd immunity, and finally how to answer an evaluate question. Check the description for my packs on command words, the adaptive specification, a revision timetable, and a template for Cornell revision notes. Hope you guys found that useful, and I'll catch you mans in a bit.